Hello friends, so today in this video we will discuss the basics of biostatistics. Uncertainties are a part and parcel of life. Medical uncertainties are an essential part of medical research. To better understand this uncertainty, let us look at an example. A group of people was diagnosed with pulmonary tuberculosis and was given treatment. All the patients in this group responded very well to this treatment. Now, if we take another group of people who have been diagnosed as pulmonary tuberculosis and given the same treatment, do you think they will behave the same way as the first group did? Probably not, because there would be variations or differences between these two groups. Let us look at what these variations can be. There could be a difference of age. Maybe one group has more children and the other group has more elderly patients. Maybe the group has differences in gender distribution. There is a difference in how males and females respond to drug. These factors are known as biological factors and include things like age and gender. Environmental factors also play a role. One group may be living in an area where there is a lot of pollution, and probably this will affect the response to treatment. Another important environmental factor is the nutritional status of the participants. If one group is significantly more malnourished than the other group, then there could be differences in response to treatment. Another difference can be because of sample fluctuations. If the sample that you have chosen is not representative of a population, then the results will be different. Another reason why this group may not respond to treatment is, as the first group did, could be because of the observer variability. The clinician may have diagnosed tuberculosis based on interpretation of some clinical signs and symptoms. This evaluation of signs and symptoms and their interpretation may vary from clinician to clinician based on experience and skill. So maybe this group has more patients with sarcoidosis, which has been misdiagnosed as pulmonary tuberculosis by an observer. The difference in response could also be because of variability in treatment strategies, and this would include differences in drug dosages, drug regimes. Another reason can be instrument or laboratory variability. The instrument which has been used to process specimens from the first group may not be the same as the one used in second group. The laboratory may be using a different technique, a different set of reagents. Incomplete information can also result in variations. Some patients may not have given a correct history because they forgot, or maybe they are trying to suppress some facts. In some communities, tuberculosis has a stigma attached to it, so they may not give the history of a contact who has pulmonary tuberculosis. Imperfect tools used to measure the response to treatment can also result in variations. All these factors can result in variations between these two groups and can result in uncertainties. So these variations results in uncertainties. And biostatistics is the science of management of these uncertainties in health and medicine. And it's a highly evolved science. What statistics does is it minimizes these uncertainties. So there are ways in biostatistics with which you can minimize these uncertainties. One of them is to have a control group. For example, if you wish to investigate a group of patients of hypertension and you want to give them drug A, and you wish to see that what is the change in their blood pressure. There could be so many factors which can affect the change in blood pressure in these group of patients. So one way is to take two groups, give the treatment to one group and do not give treatment to other group, and then you compare these two groups. So that is one way you can maybe balance out on many uncertainties which you may not be able to fathom. Another way is to match these two groups or randomize. So when you make such two groups, you have to ensure that the participants or subjects in these two groups are similar. And the other way is to randomize. So the allocation of the subjects in these two groups should be completely random. And then proper selection of subjects, as I gave an example of that canteen. If you wish to see that what is the gender of resident presenting to the canteen at 9 a.m., you have to conduct that study at 9 a.m. If a study conducted at lunchtime will not give you the same result. So selection of subjects has to be proper. And then standardized methods have to be used to minimize these uncertainties. So the instrument that you are using, maybe it's stadiometer, maybe spirometer, it has to be standardized. If it is not standardized, it will result in many uncertainties. More so, the methods that you are using. For example, if you are investigating a group of patients for induced sputum, you have to ensure that the method that you have used is standardized. Whether you are using 7% hypertonic saline, 3% hypertonic saline, normal saline, what kind of nebulizer you are using, what physiotherapy you are doing. So this will minimize variations, and more so if it has all the information, it will be easy for some other investigator to replicate this study. So as you know, in medical research, you cannot investigate the whole population. So you, out of this whole population, you choose a sample and you investigate this sample, and then you draw conclusion from this sample, and you generalize it to the overall population. So this method where you study a sample rather than whole population and investigate and draw valid conclusions from is a part of inferential statistics. So in this inferential statistics, the process of data exploration and drawing a valid conclusion is known as analysis. So in every research paper, you will see that the results section is where you see analysis, the data exploration and drawing valid conclusion from. The next part is synthesis. So in synthesis, what you do is you compare your results with those of other investigators. So in synthesis, you combine and probably reconcile results which may be in sync or results which may be contrary, and draw valid conclusions and give a holistic view of the situation. So that's about synthesis. So in a research paper, the discussion part is about synthesis. So thank you so much for watching this video. What we understand now is uncertainties are very difficult to eliminate. They are essential part of medicine and medical research. With help of biostatistical methods, you can minimize them and you can control them. Thank you.